Hi, this is a video on B2 Edexcel Biology. So we're going to go through topic 1 in this video, which mostly focuses on sort of the basics of cellular biology in terms of cells, including bacteria and a few specialised cells, microscopes, and then moving on to mitosis, meiosis, DNA, and the discovery of it as well. So let's start with cells. Cells is generally cells at this level that we're going to go through today is generally a foundation tier topic in terms of the basics, in terms of what they're actually made up of. So that's a nucleus in both plant and animal cells. So we've got an animal cell over here on the left and a plant cell here on the right. So the nucleus is in both, cytoplasm is in both, cell membrane is in both. However, a plant cell has a cell wall, a vacuole has a cell, a vacuole is also in the plant cell, and the cell are chloroplasts. You need to know the roles of each of these 6 things here. So the nucleus controls the activity of the cell. Never say it's the brain of the cell, it actually controls the activities. The cytoplasm is the site of these chemical reactions, so like respiration, that's where it takes place in the cytoplasm. And the cell membrane controls what goes in and out. So the cell wall strengthens the cell to help it keep its shape, and that's what the vacuole does as well. It's filled with cell sap so that this plant cell has this shape, and the chloroplasts are filled with chlorophyll to perform photosynthesis and therefore make food for the organism within which this is. These cells also contain mitochondria, and that is a site of respiration. So also mitochondria are present, and that's where respiration takes place. You might have ribosomes mentioned as well. They don't come up very often, if ever, and ribosomes are the site of protein synthesis. So the nucleus also contains the DNA, which is the genetic information, and you should know that for B1. Another type of cell that we need to know about is a bacterial cell. So bacterial cells have a very specialised structure to allow them to do their job particularly well. The DNA, the chromosomal DNA, is in one long loop. Is in one long looped chromosome that controls the cell's activity and replication. So bacterial cells have no nucleus, so this material is floating free in the cytoplasm. Plasmids are small loops of extra DNA. So these plasmids over here are small loops of DNA. This is the actual nuclear material. So this is the chromosomal DNA. This bit over here, the plasmid, that's sort of extra DNA, and they're not part of chromosomes. So they contain extra information. So it's like genes for like drug resistance, and it can be passed between bacteria through the plasmid. So you've got the normal chromosomal DNA, but you've got this plasmid DNA, which is sort of extra as well. And they can move between, pass information between bacteria. You also have the flagellum. So the flagellum is there. It's like a long hair-like structure. It rotates, it spins, it allows the bacteria to move. It also has a cell wall for support. Cells can be studied using a light microscope, so they've done that for a long time. Robert Hooke was one of the first people to do it. So since the 1590s, they've magnified the structure of cells. So they've been able to see the internal structure up to mitochondria, but the magnification that they can actually use isn't particularly powerful. And only since the 1930s they've been able to use more powerful microscopes. If you want to figure out the magnification, you do the length of the image divided by the length of the specimen. So however long the actual thing is, divide that by the length of the image of it that you see, the length of the image, so you divide that by the length of the image that you actually see, and that gives you magnification. So the 1930s we came across electron microscopes. They allow us to see smaller structures such as plasmids, and also examine the internal structure of cells in more detail. So they provide a greater resolution electron microscopes, therefore more detail. So let's move on to DNA. DNA, a gene is a section of DNA, and that codes for a specific protein. So DNA is 2 strands coiled together to form a double helix. The strands are linked by complementary bases. We'll go through what the complementary bases are in a second, and they're joined together by weak hydrogen bonds. So the complementary bases, we have A and T, that link with the C and G. Make sure you can spell the names of those correctly as well. So at this point you pause the video and link the letters with the names and make sure you can spell them correctly. It has come up every now and again as a multiple choice question, and always a 1-mark question. You should have done an experiment in class about extracting DNA from, say, kiwi fruit or strawberries. Okay, it's pretty straightforward. You need to make a solution to break up all the cells within the fruit and dissolve the membrane around the nucleus that releases the DNA. You add salt and washing up detergent together in a beaker. That makes you don't want to foam this detergent. Essentially, it's creating a buffer. You put the beaker, which contains the chopped up fruit with the solution, and you put that in a water bath at about 6 degrees. This denatures the enzymes that could digest the DNA, so the DNA is not going to get digested now because the enzyme has been denatured, and we'll talk more about denaturing enzymes in a bit. In another video. Then you cool the mixture down. You prefer to do it to the ice cold, but that's quite hard to do. You mix the solution to break up more cells. You filter the mixture. This removes large parts of the cell. You add ice cold ethanol, and the DNA will come out of the solution, just a stringy white substance that can be fished out using a bent paper or glass rod. So how was DNA actually discovered? Of course they wouldn't have discovered it using an experiment like that. There's 4 people you need to know about that are important in the discovery of DNA. There's Watson, Crick, Franklin and Wilkins. Rosalind Franklin and Maurice Wilkins worked out that DNA had a helix structure by direct use of X-rays onto crystallized DNA. They looked at the patterns that were formed by the X-rays, which gave them an insight into a structure. Watson and Crick used this idea along with the knowledge that the amount of A and G matched the amount of T and C to make a model of DNA. Okay, so they looked at the actual complement. They came up with the idea of complementary bases by looking at the amounts of A, T, C, and G. So after all this understanding of DNA came about, the HGP came in, which was essentially a huge mapping program. It was the Human Genome Project. So genetic information in an organism is called a genome. The HGP started at the end of last century, so late 90s, and it's really ambitious. It wanted to work out the sequence of all of the 3 billion base pairs in the human genome. It wanted to identify all the genes and develop faster methods for actually sequencing DNA. It was finished in 2001, but work still continues to identify all of the genes in the human genome. They use DNA from several people to get a sort of average sequence, so the sequence that's there in everyone, but each person has a unique sequence, unless they're identical twins. It's also allowed scientists to find out what all of the genes do, which is brilliant. So the pros of this is that it's helped scientists to predict and prevent some genetic diseases, develop better medicines, improve diagnosis, improve forensic science. The cons are that it could increase some stress in people because they could know that they're going to develop a condition in quite an early age, and that could cause anxiety. In terms of where we have racism, we could potentially have genism as well, where some people are forced not to have babies because they know that they're going to have a genetic condition. This could lead to discrimination, possibly by insurers. They won't give certain people insurance, but also it might be that employers don't employ certain people. So another thing that's come out of this whole understanding of DNA is genetic engineering. Genetic engineering is also called GM, or genetic modification. It's not the same idea as cloning. Please differentiate between the two. Cloning techniques are used in genetic engineering, but don't confuse them. So here's a table to help you distinguish between the two. Cloning produces exact copies, and the genes are copied within the same species. Genetic engineering produces a unique set of genes, so it's not a copy, it's a unique set, and they actually swap genes between species most of the time. So how does genetic modification actually work? Well, let's highlight that up. So certain enzymes cut pieces of DNA from a particular gene from one organism. They join it into a gap of DNA in another organism. So they're making. So this means that the new organism with the inserted genes has genetic information from one or more new characteristics. So it's going to have one or more new characteristics now because it's got new information in it. So it might be able to produce a useful substance like insulin for diabetics, or it might be able to carry out a new function, something it couldn't do before. So that organism has now been genetically modified. So here are some of the ways in which it could benefit humans. Beta-carotene in golden rice helps to reduce the vitamin A deficiency. So vitamin A deficiencies are big problems in South Asia and Africa. It could actually lead to blindness if they're not careful. So golden rice contains 2 genes from other organisms that together enable the rice to produce beta-carotene, and that means it can actually reduce vitamin A deficiencies. Human insulin, as I just mentioned, can be made by modified bacteria. So they insert the human insulin gene into bacteria. That means that insulin is produced quickly and cheaply. So that's what's in this diagram over here as well. Herbicide-resistant crops. So say you want crops to grow, but you want to spray herbicide on your farm to kill out the weeds. Okay, you can do that by making herbicide-resistant crops. So it increases yield by making sure there's less weeds there, and also makes more food. But there's some long-term side effects that we might not be aware of completely yet. So sometimes people don't know what these long-term effects could be. They could be dangerous. If you're killing weeds and flowers on farmland, that could affect biodiversity. You're killing species that could have harmful effects. We don't know the effects of that. Some people think that allergies might have been adapted to, might have come about, to certain foods that have been genetically modified, although there's no proof of that yet. There's no link for that proven yet. But a big concern is that transplanted gene that's in these crops may somehow get into the environment and into the weeds. Now that could create super weeds, in other words, weeds that are resistant to herbicides that you can't kill anymore. That could be very dangerous. So once you have like one of these cells that's got a specific characteristic, how does it actually reproduce? How does it actually replicate itself? So that's what we're getting onto here in terms of cell division. There are 2 types of cell division that you need to know about. The first one of these is called mitosis, and the second one is called meiosis, or meiosis, as some people call it. First, I need to make sure you understand 2 terms. The haploid number, that's 23 chromosomes. The diploid number, that's 46 chromosomes. We, as humans, have 23 pairs of chromosomes, so we have 46 chromosomes. So haploid gametes, so like a sperm or an egg, will have 23 chromosomes. Mitosis is the production of 2 daughter cells, so 2 new cells, with identical sets of chromosomes in the nucleus to the parent cell. So whatever it started with, the 2 new cells have identical sets of chromosomes. This results in the formation of 2 genetically identical diploid body cells. So if they're the body cell, 46 chromosomes. Okay, so it occurs during growth, repair, and asexual reproduction. Cloning is an example of asexual reproduction that produces genetically identical copies. So you start with the parent cell, the DNA replicates. You can see it's got twice as much of it now, and then it splits up into 2 daughter cells to make exact copies of itself. Very straightforward for mitosis. Meiosis or meiosis, that produces 4 daughter cells. Each has half the number of chromosomes. So in other words, they have the haploid number. They have 23 chromosomes. So this means that it can actually form genetically different haploid gametes. So there's a parent cell, it replicates, it splits into 2 daughter cells. Okay, but instead of each DNA splitting and making identical copies, each sort of section, each sort of cross, if you see what I mean, comes out, and then they split apart. So actually these are different now. So cloning. What actually happens in cloning? There are several stages in the cloning process that you need to know about. The first is you remove a diploid nucleus from body cells. That's for all 46 chromosomes, all 23 pairs. You remove the nucleus of an egg cell, enucleation. So you're removing the nucleus from an egg cell. You insert the nucleus you got from the body cell into the enucleated egg cell. So where there was that gap, they're now putting in the body cell, the nucleus from the body cell. You stimulate that cell to make it divide by mitosis, and then you implant that into surrogate mammals. That surrogate will now allow the cells to reproduce inside of it and foster it, and essentially it will grow and become a clone. So it can deal with situations such as shortage of organs for transplants or organs for animals, and it helps the scientists understand how embryos actually develop and the aging of age-related disorders. Clones can also sort of preserve endangered species. However, it could reduce the gene pool. So if you've got loads of clones, they've all got the same DNA, there's not going to be as much variation. Now, if there isn't as much variation, you don't get evolution in the same way, and you also don't get people dealing with disease in the way they normally would. So actually, simple diseases could become a lot more difficult to deal with. There's an implication that cloned animals might not live as long. Dolly the sheep only lived for 6 years, the very first cloned sheep. This hasn't actually been confirmed yet, so they need to do further tests to see the link between premature death and cloning animals. So sometimes, well, quite often actually, the cloning process fails, and some clones are born with genetic defects, and some clones have poor immune systems, so they suffer from more diseases. So there's lots of difficulties with cloning, yet we haven't nailed it. So it's not something that we're completely happy with. Stem cells is another big area of research right now. Essentially, they're cells that are found in the embryo and are unspecialized. So this means they can become any other type of cell. So they can become a red blood cell, they can become a cell in the arm, they can become a cell in the eye. They're undifferentiated. So when they grow into a type of cell, they differentiate. So in animals, stem cells aren't continually produced into adulthood. Okay, so this is why we use embryonic stem cells for research. They have to come from an embryo. There's a lot of controversy over this. There's a lot of ethical issues. So remember, there's no sort of right or wrong answer, but there's, who should make a decision on this is very, very difficult to discuss. So, stem cells come from aborted embryos right now, or spare embryos from fertility treatment. So some people think this is unethical, because a lot of people think that it's destroying the life purely for research purposes. But the problem is that scientists believe we could actually use these cells to cure diseases such as Parkinson's disease, or rebuild bones and cartilage and repair the immune system if it's become damaged. The current legal position is that stem cell research is legal to an extent, but many want this to change. And under the previous government in America, one of the previous governments in America, it actually became completely illegal. Stem cell research was completely illegal for a while. With plants, however, stem cells are being constantly produced. In an animal embryo, the stem cells are often produced and then go on to. In an animal embryo, the stem cells are often produced and then go on to develop and differentiate into specialized cells. So they can only reproduce that specific cell again. They can't become a stem cell again. When they replicate, they just replicate whatever they are now. In plants, only the stem cells are ever produced, and then they travel to specific places in the plant and they differentiate once they're there. So the mitosis actually happens on the stem cells in that one. So big topical area, a lot of people worry about that one. Right, protein synthesis. Very, very difficult part of B2, and it's one of the last bits in B2 topic 1. Apologies, I thought enzymes was in the next topic, it actually isn't this topic. Right, so protein synthesis is what it says in the tin. It's making proteins. So proteins are made by cells by stringing together something called amino acids, and they put these amino acids in a specific order, and then that protein can do a job, essentially. So there's only 20 amino acids, and those 20 amino acids can make up of thousands of different proteins. The order in which the bases in a gene are put together essentially tells the cells what order to put the amino acids in. So a triplet, or three bases, so triplet codes for an amino acid. So 3 bases pairs together code for an amino acid. DNA determines which genes are switched on or off, and therefore which proteins the cell produces. It's pretty straightforward. Proteins are made by ribosomes, as we said before, in the cell, and DNA can't move out of the nucleus because it's too big, and it needs to get information to ribosomes in the cytoplasm so that a protein can be made. So it uses something called a messenger, and it calls that messenger mRNA, so messenger RNA, which is like DNA, but it's only a single strand of it. So this is what it does. 2 strands of DNA unzip, so you have your double helix, it unzips, and 1 strand is used as a template to make the mRNA. The base pairing ensures that it's complementary, so that's called transcription. It says, here I am, this is what I look like, make a copy of me. That copy is called mRNA. That mRNA molecule then moves out of the nucleus, I'm down over here now, and joins with the ribosomes. Okay, so it comes out and meets these ribosomes. The amino acids that match the mRNA code are brought to the ribosomes by tRNA, so it's called transfer RNA. The job of the ribosomes is to stick amino acids together in a chain to make polypeptide, which is a protein. This follows the order of the triplet bases in the mRNA. That's called translation. So the key phrases are transcription and translation. Transcription is where you're making mRNA, it's that exact copy. Translation is when you're making the new DNA essentially. So this is where you've got the order of the triplet bases in the mRNA being replicated. So this results in each protein of a specific number and sequence of amino acids. So this allows a protein to fold into the right shape to do its specific job as a specific enzyme, possibly. Sometimes mutations can happen in this process. They can be harmful, beneficial, or they can do nothing. A harmful mutation causes genetic order, like cystic fibrosis. Beneficial mutation could cause a new characteristic, which could be beneficial to the organism. Think about natural selection, survival of the fittest, back in B1. A neutral mutation probably does nothing. It's not beneficial or harmful. So enzymes are biological catalysts. So enzymes link to all of this because enzymes are proteins as well. So they're biological catalysts. They speed up chemical reactions occurring inside and outside cells, including this replication of DNA, protein synthesis, and digestion. Enzymes are also proteins, as I just mentioned, but then they're folded into complex shapes that allow smaller molecules, substrates, to fit into them. The place where the substrate molecules, things that actually fit into the enzymes, fit is called the active site. So this is the active site, this is the enzyme, and here's a substrate that can fit into it. This leads to the idea of like a lock and key. So this is like the key, this is the lock, and they fit together. Only certain enzymes fit certain substrates. Okay, so it's a one-to-one relationship essentially. If somehow the shape of this enzyme changes, then its active site might not work anymore. Okay, we call that idea denaturing, and that can be affected by high temperatures or extreme pH. It's wrong to say the enzyme has been killed. Although they're made by living things, they're not actually alive themselves, they're proteins. So what factors can affect how quickly enzymes what factors can actually affect the activity of enzymes? So as temperatures increase, so does the rate of reaction. But at high temperatures, these enzymes denature, so they change shape, so therefore enzyme activity doesn't take place. The graph shows a typical change in enzyme activity with increasing temperature. So it usually increases to an optimum point and then it decreases. The optimum point can vary between different enzymes. If it's inside the body, it's usually about 37–40 degrees Celsius. And in pH, again, that optimum point can vary. It increases to its optimum point and then decreases again as it denatures. Okay, but that optimum pH can be anywhere depending upon the situation and what we're using for. So intestinal enzymes generally have an optimal pH of about 7.5. However, enzymes in the stomach have an optimal pH of about 2 because in the stomach, it's very acidic conditions. Substrate concentration has an effect on rate of reaction up to a point, but then there's a point where actually you can have as many substrates as you want, but there's not enough enzymes to actually deal with all those substrates. That's what happens here. Concentration no longer has an effect because the fact is saturated. So this is the point of saturation. All the enzymes are currently doing their job and you can't get to any more enzymes quicker. So you can actually investigate the rate of reaction using amylase as the enzyme and starch as a substrate. So iodine has been used to test for the presence of starch. Once the starch is no longer present, the iodine will stop turning dark. So the experiment can be repeated with varying temperatures or pHs, whatever you're varying, and you can see that sometimes the reaction hasn't occurred because of the fact that to stop turning dark, you can see it stop turning dark over here because the enzymes become denatured. So iodine spot test for starch, 1 minute intervals for a starch amylase mixture kept at 37 degrees C. Excellent, and that's the end of topic 1 on B2 Edexcel Biology.